Dear students, Model 4 Pedigree Analysis, History, Symbols used in pedigree studies, pedigree construction and pedigree of autosomal inheritance. The main objectives of this episode are to know about the history of pedigree studies, to understand the symbols of pedigree analysis, to understand the construction of a pedigree, to understand the pedigree of autosomal inheritance. Introduction. Pedigree is constructed to establish the mode of inheritance of a character in a family. The pedigree shows the presence or absence of a character as it relates to the relationship among parents, offspring and siblings. Pedigrees present the process of interpretation of information displayed as a family tree. Pedigree is constructed using a standard set of symbols which gives information about the disease status of each individual. If a single individual is affected within the family, then the pedigree cannot explain a particular mode of inheritance and it is difficult to distinguish from non-inherited conditions. If more than one individual is affected, then the pattern may provide substantial proof of the mode of inheritance. Hence, in this episode, the following aspects are going to be emphasized. A. History. B. Pedigree chart and symbols. C. Pedigree construction. D. Pedigree of autosomal inheritance. The word pedigree first appeared in the English language in 1410 and introduced in 1828 to name the tree of an animal. In France, the pedigrees were written by the breeder himself by hand and certifying that the information is written the same as once found in the breed stock book. In the UK, pedigrees are recorded officially in the College of Arms, which has records from the 17th and 18th century. Pedigrees continue to be registered at the College of Arms and kept up to date voluntarily and are accessible to the general public with payment of a fee. A diagram of pedigree predicts the biological relationships between individual and their ancestors looking into family history. Popularly, pedigree is used for different animals such as dog and horses to look at the transmission of genetic disorders. Pedigree is commonly used in human families to determine the mode of inheritance of particular diseases. Therefore, pedigree analysis is an essential tool in both basic research and genetic counseling. Family history is a crucial information of an individual's past and future life, which can be used as a diagnostic tool. This will help to make decisions about genetic testing for the patient and to calculate risks for the family members. Accurate family history will be essential to establish a pattern of transmission. Family history can also identify potential family problems such as heart disease, diabetes or cancer, where an individual with increased risk for the future. Early identification of increased risk may allow the individuals and health professionals to take steps to reduce risk by implementing lifestyle changes, introducing medical interventions and or arising disease surveillance. Therefore, a genetic disorder cannot rule out in the absence of a history of a family. When patients give information about their family history, a pedigree can be created faster than collecting the information in writing and allows patterns of a disease to emerge as the pedigree is drawn. Since family history is continually changing, the pedigree can be updated in future visits. Patients are allowed to record information and update their family history regularly. Therefore, the pedigree is a roadmap used to do some detective work and is often used to study the genetics of inherited diseases. Geneticists use a set of symbols to represent an individual's sex, phenotype and family relationships. Males are represented by square, while circle represent females. Horizontal line connect parents, while vertical lines make to the symbol for their offspring coming from horizontal lines. The Roman number 1 is used to indicate for the first generation, 2 for the children of the first generation and 3 for the grandchildren. The affected individual of the family is called as proband or propositus. If an individual has to have symptoms of the disease, then the symbol is filled in. Sometimes a half filled in a symbol is used to indicate a known carrier of the disease. In this condition, he or she does not have any symptoms of the disease. Since they are heterozygous, they pass the disease on to subsequent generations. Note that when a pedigree is constructed, it is often unknown whether a particular individual is a carrier or not. So, not all carriers are always explicitly indicated in a pedigree. A line joining a male and female will be called as mating, while a consanguineous mating is with 2 lines. Each pedigree chart gives information about the inheritance of a single trait within a family. These diagrams help to visualize relationships within families, particularly large extended families. Pedigree determines the mode of inheritance of dominant and recessive condition of genetic diseases. Therefore, the pedigree chart is drawn using factual information. However, there is always some possibilities of errors in this information, primarily when relying on family members, recollections or even clinical diagnosis. The symbols used in pedigree charts are according to recent recommendations made by professionals in the field of genetic counseling. Generally, necessary information on family history includes 3 generations. To begin taking a family history, healthcare professionals ask the patient about his or her health history and then ask about siblings and parents. Questions should include 1. General information such as names and birth dates. 2. Family's origin or racial or ethnic background. 3. Health status including medical conditions and ages at diagnosis. 4. What is the age at death and cause of death of each deceased family members. 5. The outcome of pedigree of the patient and their relatives. First, it has to list all the members of the nuclear family, then collect the health status of all other members. After recording the family history of the patient's closest relatives, then enter one generation at a time and ask about aunts, uncles, grandparents and first cousins. Pedigree analysis can determine genotypes, identify phenotypes and predict how a trait will be passed on in the future. This information from a pedigree makes it possible to determine how individual alleles are inherited, whether they are dominant, recessive, autosomal or sex linked. The modes of inheritance reflects whether a trait is dominant or recessive and the chromosomal linkage of the character in question. Penetrance and expressivity are complete if the trait displayed in the pedigree is due to the action of a single gene. If the character is dominant, one of the parents has a character. Dominant traits present in every generation. If the trait is recessive, none of the parents is required to have the character because of the heterozygous condition. Females are less commonly affected than males in X-linked recessive traits. In autosomal traits, both males and females are usually in equal proportions. There are 6 standard modes of inheritance that can be reliably determined by careful pedigree analysis. They are autosomal dominant or recessive, sex-linked dominant or recessive, Y-linked and mitochondrial inheritance. For autosomal dominant and X-linked dominant, there is no question of a carrier, since the affected allele will be dominant. For autosomal recessive cases, it is a little tough to figure out the carrier without sufficient information. It is challenging to distinguish homozygous dominant from heterozygous individuals who are unaffected. However, we can tell if the affected individual is homozygous recessive. The exceptions will be when one parent is homozygous recessive, and then we can say that offsprings will be carriers of the condition. For X-linked recessive, the carrier will always be the mother or female. Since she has two X chromosomes, one can compensate for the function of the other non-functional chromosome. The father or male can never be the carrier because he has only one X chromosome. If affected, the condition will be indicated in the pedigree chart. The construction of pedigree is by trial and error. We consider one mode of inheritance at a time for each mating in pedigree and try to find evidence against it. One has to repeat for each pattern of inheritance, that is autosomal recessive or dominant, sex chromosome recessive or dominant. Construction of pedigree is an assumption, that is, for rare traits, unaffected people entering into the family pedigree, that is, by marriage, are considered homozygous normal. Pedigrees can frequently rule out, but not necessarily prove a particular mode of inheritance. Problems in constructing a pedigree: poor medical records, scattering of family members, inaccurate information, miscarriages and stillbirths, concealed adoptions, variable expressivity of genotype as phenotype, incomplete penetrance of genotype showing phenotype. Pedigree can frequently rule out, but not necessarily prove a particular mode of inheritance. Now, let us learn the pedigree involved in the autosomal patterns of inheritance. This mode of inheritance helps to understand the inheritance of autosomal traits in both the sexes equally. Further, it can be classified into two types: autosomal dominant inheritance and autosomal recessive inheritance. Let us understand the patterns of autosomal dominant inheritance. In this inheritance, only one copy of a disease allele is necessary for an individual to be susceptible to expressing the phenotype. Since the transmission from parents to offspring, this is called vertical inheritance. The portions of affected males and females are equal. In this case, a 50% chance of the offspring will inherit the disease allele in each pregnancy. Male to male transmission can be observed. Traits should not skip generations. A normal person married to an affected person should have 50% of the offspring being affected. It also indicates that the affected individual is heterozygous. In autosomal dominant condition, an affected parent passes half of the trait to their children. However, one has to be careful that we may not see 50-50 chance of inheritance the causative allele if the progeny is very less. Therefore, in any autosomal dominant condition, a person can have several children without the dominant allele. This is possible like flip a coin 5 times and get heads every time. If a new mutation has occurred, all affected individuals will have at least one parent who carries the disease allele. Some of the examples are myotonic muscular dystrophy and Huntington disease. Now, let us understand the patterns of autosomal recessive inheritance. In this condition, 2 copies of disease alleles are required for expressing the phenotype. Generally, the parents of an affected individual are the gene carriers and are not affected. In every pregnancy of carrier parents, the offspring will have a 25% chance to inherit 2 copies of the disease allele and have the phenotype. In this case, the family will have a 50% opportunity to inherit a disease allele, and thus they are the carriers. About 25% of the offspring will not inherit the disease allele, and hence they will not express the phenotype. These offspring will not pass the disordered genes to their offspring. As like autosomal dominant inheritance, the proportion of affected males should be equal to the proportion of affected females in a given population. A trait often skips generation. Distribution of the trait is generally close to equal distribution among the sexes. Traits have found in pedigrees with consanguineous marriages. Most affected individuals have normal parents. When an affected person is married to a normal individual, all of the children are normal, suggesting the normal parent is homozygous dominant. Examples are sickle cell anemia and cystic fibrosis. Inbreeding, that is mating between relatives, increases the chance that mating will be between 2 heterozygotes. Thus, matings between relatives run a higher risk of producing abnormal homozygous recessives than do mating between non-relatives. It is for this reason that first cousin marriages are responsible for a large portion of recessive disease in human populations. It is seen more in Karnataka and Andhra Pradesh. Now, let us look at a pedigree of patients with phenylketonuria, a rare metabolic disease. What is the type of inheritance of this disorder? What do you think, is it recessive, dominant or neither? In this condition, it appears to be recessive. However, we cannot rule out that it is due to the complex type of inheritance. In a recessive case, it takes two copies of the affected allele to cause the disease. That means, one inherited from the father and one inherited from the mother. Therefore, all the individual that inherit an autosomal recessive conditions have unaffected parents. Both of these unaffected parents will have a copy of the recessive allele that will make them carrier of this disorder. Always a carrier has one copy of the recessive allele and does not exhibit the trait. Generally, in a pedigree, carriers can be designated by placing a spot in the open circle or square. Relationship of affected individual infer some carriers of the disease. However, with advances of present day knowledge on human genetics, it is now possible to conduct genetic testing on individual and find out that they carry the defective recessive allele or not. Since both the parents are carriers for phenylketonuria, each of her siblings has a 25% chance of inheriting the disease condition and 50% chance of inheriting one copy with carrier condition. Thus, nephews and nieces of the proband may inherit phenylketonuria if their parents happens to be a carrier. Now, do you think phenylketonuria is autosomal or sex linked? You are correct if you say autosomal. Sex linked conditions usually affect males since they have only one copy of the X chromosome. In contrast, females have to inherit two copies of the causative allele. In the pedigree, we have one male and one female have phenylketonuria. So, this is the autosomal condition. Autosomal recessive conditions are challenging to identify from pedigrees alone because the disease often skips more than one generation. Besides, affected individuals inherit copies of recessive genes from both parents. Even if both the parents are carriers, only one out of every 4 of their children is possible to be affected. Therefore, sometimes autosomal recessive disorders can be identified from pedigree alone if the pedigree is enormous. In families where parents have lots of children, for example, 8 or more, the 1 in 4 ratio of inheritance of autosomal recessive condition from 2 carrier parents might become more identifiable. Besides, long family history also increases the chances of identifying a rare autosomal recessive disease that makes a prominent appearance in a family every few generations. To conclude, the construction of pedigree is an important step in understanding genetic diseases. It is a roadmap for the diagnosis of a trait. In the next episode, let us study other types of pedigrees. Thank you.